Hello everyone. Today we are going to discuss about how we can measure radioactivity. Last previous two lectures we were describing about radio labeling techniques, what are radioisotopes, what are the major radioisotopes used in biological sciences, how exactly radio labeling is useful, and what are the different types of radioisotopes that are used frequently in biology. So this was what we were discussing, and we also discussed about what are the different types of radiations that these radioisotopes could probably emit, and what are the levels of penetration these radioactive radiations could have. So today we are going to discuss on how exactly we can measure this radioactivity or the radiation that is being emitted by the radioactive isotopes. You must all have heard about Marie Curie and Pierre Curie. So these were two legends who were behind this radioactivity, especially Marie Curie. It was her research on radioactivity and radiations that has led us to what we know about radioactivity today. So, with that, uh, let's see how exactly radioactivity affects certain substances and how we can measure radioactivity. So, radioactivity, as you all know, is a property of an unstable isotope which keeps emitting energetic particles in form of rays from its nucleus, and this amount of what do we say, the emitting energy and the amount of radioactive material is measured by how many nuclear decay is taking place per second, and this is known as the activity, and it is usually measured in units called as Curie, and it is denoted with a capital C and a small I. And Marie Curie and Pierre Curie, and as I said earlier, they shared Nobel Prize for the discovery of this radioactivity. And interestingly, Marie Curie is the first woman scientist to have received Nobel Prize. And also, she is one person, or maybe the only person, who has got Nobel Prize in two different fields, physics and chemistry. And she belongs to a family where there are five Nobel Prize winners. And it will be interesting that you go and read about Marie Curie and her experiments with radioactivity. And unfortunately, because of all these overexposure to radiations, she died very early because of some disease that had its origin because of the overexposure to radiation. So that is one side. We will be discussing about the hazards of radioactivity and how we can handle it in another session. So coming back to the measurement of radioactivity, as we mentioned earlier, there are four basic types of radiation: alpha, beta, gamma, and neutrons. And these are the radiations that are emitted during the decay or disintegration of the radioactive isotopes. And these isotopes can be emitted alone or in combination with one another. So, the unit of radioactivity, as I said, is measured, or the radioactivity is measured in terms of disintegrations of the radioactive isotope happening per minutes or per second. You could say disintegrations per second or disintegrations per minute. Curie is recognized as a unit for radioactivity. One Curie is equivalent to disintegration per minute or disintegration per second produced by 1 gram of pure radium 226. 1 Curie is equivalent to 2.2 into 10 raised to 12 disintegration per second, or 3.7 into 10 raised to 10 disintegration per second. Sorry, the earlier one was disintegration per minute. Then but the international system of units recognizes becquerel, or it recommends becquerel, the term becquerel, for the unit of radioactivity. And this becquerel is equivalent to 1 disintegration per second. That means 1 Curie is equal to 3.7 into 10 raised to 10 becquerels. So becquerel is now the accepted international system of units for radioactivity. Radioactivity can be measured using certain instruments, and what type of instrument is to be selected for detection and measurement of radioisotopes depends on the type and the energy of the emitted radiation. So, what is the kind of radiation or rays that is getting emitted from the radioisotope? Accordingly, we choose instruments for the detection and measurement. For example, most radioactive isotopes, such as selenium 75, emit gamma rays, but there are some exceptions, such as tritium, carbon 14, phosphorus 32, sulfur 35, and calcium 45. They are known as beta emitters because mainly when they decay, they emit beta radiations. Likewise, gamma radiations or beta radiations, which are emitting from the radiolabels or the radionuclides, these are detected using two different systems, or we could say that a combination of a scintillator and a photomultiplier tube. Uh, Also, depending on the type of radiation that is being emitted, we can use another instrument known as Gigamuller counter. And then, when we are dealing more with the biological molecules like DNA, RNA, and proteins, we use autoradiography as well. So, we will be discussing about all these methodologies and all these instruments in detail. So these are the most common detectors which are used for detecting and measuring radioisotopes in biology. As I mentioned earlier, Gigamuller counter, it is primarily employed to study the emitting beta particles of intermediate to high range, high energy range. And this may also be used at low efficiency for the measurement of gamma radiations as well. Then comes the solid scintillation counters, which are generally employed with gamma ray emitting isotopes, and liquid scintillation counters are used with isotopes emitting low energy beta particles. So, Gigamuller counter, solid scintillation counters, liquid scintillation counters, and other than the scintillation counters, we also make use of autoradiography, which we'll be discussing later. So, Giga counter, as they are commonly called, the Gigamuller counter. It is an electrical device that helps to detect various type of ionizing radiations. You can see here the two personalities behind this Gigamuller counter, Hans Giger and his student Walter Muller. So they were behind this Gigamuller counter or Giga counter, and they received they are known to have devised this equipment for detecting the different types of ionizing radiations. So, Giga counter is now widely used in applications such as radiation dosimetry, radiological protection, experimental physics, as well as widely used in nuclear industry for detecting ionizing radiations like alpha, beta particles, neutrons, as well as gamma rays. So this is Gigamuller counter here. Now let's see how exactly this instrument works. If we say about the constitution of the Gigamuller tube or Gigamuller counter, it has got a cylindrical tube-like thing inside which you can have a central wire or an anode, and this is your metal tube which functions as a cathode, and in this tube will be fixed a low pressure inert gas like argon. Then there is a stop window made up of thin mica, and then you have your rate meter, which helps us count the number of pulses that is being generated during this radioactivity. So, Giga counter consists of a gigamuller tube, then the sensing element that detects the radiation, and the processing electronics, which helps to display the result. And these can detect alpha, beta, as well as the gamma rays. So the working is somewhat like this. the tube is filled with low energy or low pressure gas like argon, and the end of the tube is sealed with a mica window, and this is thin enough in order to allow alpha particles to enter the tube, as well as it let go beta and gamma radiation. So when we are trying to count the radiation or detect the radiation that is being emitted from a radioisotope, what happens? This, suppose this is your radioisotope or that molecule which is labeled with this radio labeling, it starts emitting radiation. And this radiation, once it enters into this gigamolar tube, what happens? It goes and reacts with the argon gas that is present inside. So once these gamma radiations enter the tube, the argon gas gets ionized. Once the argon gas is ionized, there happens a whole avalanche of ions between the electrodes, because many more number of uh, molecules are getting ionized, and this creates a current inside this tube, and the gas conducts and a pulse of current flows into the circuit. So once there is a current detected by the circuit, what happens? It is either getting scaled up or it goes to the rate meter. A scaler helps to count the pulses and show the total on the display. Likewise, the rate meter indicates the number of pulses or counts per second, and the complete apparatus is now called as the Giga counter or the Gigamuller counter. So what is happening here? When radiation enters the Gigamuller tube, the gas inside the tube, for example argon, it gets ionized, and once it is ionized, there is an avalanche of ions that is being formed within this tube, which creates an electrical pulse. This electrical pulse is sensed by the electrodes, and that is getting passed on to the rate meter, which can measure exactly how many pulses or how many counts are coming per second, and this is a kind of measurement of radioactivity. So this is how the Gigamuller counter works in detecting radiation. The second major method for detecting and measuring radioactivity is the scintillation counter. And scintillation counters help measure radioactivity in liquid samples as well, and also on samples on strips of filter paper. The instrument that we are using is the scintillation counting method. But when we have samples, for example, a DNA or an RNA molecule, which is present on an agarose gel, and suppose the scientists want to find out whether a particular piece of DNA or a gene is isolated or not, or maybe a PCR product, we want to know whether our gene of interest got amplified or not, whether the transformation has taken place or not. In such conditions, when we have a sample on an agarose gel, we could either go for hybridization with a radioactive probe, and we need to detect it. In this case, the detecting agent or the detecting mechanism that we are making use of is autoradiography, which helps us to pinpoint the location of the radioactive bands or the spots, thereby helping us identify our gene of interest or whether the transformation has taken place or not. And such details we get from autoradiography. We will check out one by one. Let's take a look at how scintillation counting works. Scintillation counting mainly relies on certain chemicals called the scintillants. The speciality about the scintillants are that they can emit a flash of light when they absorb high energy molecules, like the beta particles. So, once the beta particles or high energy radiations fall on this chemical known as a scintillant, they will emit a flash of light. So, these flashes of light are the beta particles that are released by the radioactive isotope result in emission of the flash of light. And this flash of light is being sensed by a photocathode, which passes on this impulse into the scintillation counter, and within the scintillation counter, these ions are, or these impulses are getting magnified with the help of a photomultiplier tube. So from the scintillation scintillator, the flash of light or the photons that they emit get transferred to, or to the photocathode, which lets it inside or into the focuses into another tube like structure known as a photomultiplier. The speciality about this photomultiplier is, if you can look carefully in this picture, there are certain electrode like things that are known as the diodes. The speciality about these diodes is, once this photon comes and strikes on this diode, it multiplies it, then it comes here, then again it is getting multiplied, and with each transfer, the number of photons keeps on increasing. And once it reaches the other electrode, which is then connected to a detector or instrument which can read and display the number of photons, that is how the scintillation counter or counting is used for identifying or to measure or detect the radiation. So what happens here, when the high energy photon falls on this scintillator, it starts emitting low energy photons. These low energy photons are transferred onto the photocathode, which directs it into the photomultiplier tube. Within the photomultiplier tube, we have multiple number of diodes. With each diode, the number of photons keeps on increasing or keeps on multiplying. And once it reaches the anode, this impulse is being connected to the recorder, which helps us to display the number of photons, or the helps us measure the radiation that is being emitted. So this is exactly how the scintillation counter is being used to measure radioactivity. As you can see here, this is your sample, and here you can see DNA, and with the radioactive labeling, and this is your scintillator, and in this case this is kind of the liquid scintillation system, where you have your sample in the liquid form. Okay, so once the beta particle is getting emitted, okay, what happens is it will give a flash of light. As you can see here, a light pulse. These light pulses are then transferred into the photo multiplier tube or the photo cell, which will detect it. And finally, on the counter, you can see the count of the photons that are being emitted per for a particular period of time. Okay, so this is how it works. Then liquid scintillation counter is a standard laboratory method to quantify the radioactivity of low energy radioisotopes, especially the beta emitting and alpha emitting isotopes. And this is the instrument that you can see over here. The light sensitive liquid scintillation counting system, or the method, it requires a specific scintillator, or it requires two specific cocktails of substances which helps to absorb the energy, which can be detected as light pulses, and they mainly include aromatic organic solvents and scintillators or flows, as we call it. And this is a schematic representation of how scintillation process works in liquid scintillation counting. So this is your radioactive molecule, which is emitting the low energy beta particles. And once this beta particle falls on the solvent system, what happens? It will start emitting a pulse of light. This pulse of light will fall on the fluor molecule or the scintillator. So once this solvent molecule is getting excited, it again sends off energy molecules again. And what happens is these electrons of the energy molecules are falling on the scintillators, which now responds by emitting photons. And these photons, as we saw earlier, will enter into the photomultiplier tube where it gets multiplied, and the counter will count the number of pulses. So as you can see here, this is a real uh, what is happening within this system. You have your beta particles are emitted, and this causes the solvent to become excited. And once this energy from the solvent system is passed on, what happens? The fluor or the scintillator will start emitting light or the photons. And this light enters into the photomultiplier tube, and then it is counted by the counter. So this is about the liquid scintillation counting. So we spoke about Gigamuller counter, and now we have seen how scintillation counting is used to detect and measure radiation. So radioisotopes in biology can be detected using many different systems. We have discussed about scintillation system as well as the Gigamuller system. And again, with isotopic labeling, there are different ways of detecting the presence of isotopes. For example, they can be detected based on their mass, the mode of vibration they produce, and the type of radiation they emit. So mass spectrometry is one instrument which helps us detect the mass differences, while the infrared spectroscopy, we have already discussed about this, it detects the difference in vibration mode of the particles within. And the magnetic nuclear resonance detects atoms with different gyromagnetic ratios, and also radioactivity can be detected by an ionization chamber or even by gel autoradiography. So isotopes which are used in biology can be detected using many different modes, taking into consideration the characteristics of that particular compound or the molecule. We can make use of their differences in the mass, and then we can use mass spectrometry. We can relay on the mode of vibration of molecules when the beta particles, the alpha particles, or the radiations fall on them. And in this case, we will be making use of infrared spectrometry. We can even use the magnetic properties of these molecules, and in that case, we will be using the magnetic nuclear resonance system. Then now we will come on to autoradiography. As I said earlier, when we are using DNA molecules, when it is present in a gel, we make use of what we call as autoradiography. So we will now discuss about the principal mechanism and the uses of autoradiography. Autoradiography is a method which allows the detection or localization of radioactive isotopes in biological system. This helps to demonstrate the distribution of radio-labeled compounds within an organism or a cell. It also helps to identify where exactly these compounds are incorporated when we are labeling it, uh, where exactly it is present within a cell or a tissue. It helps us reveal the location of the final product of a process when we are using a radioactive precursor, and we can find out where exactly the compound is, since the metabolism uses this radioactive precursor. The mechanism of autoradiography works like this. We incubate the tissue with a radioactive ligand. Here, it can be a tissue or it can be a gel, it can be a blot. What happens? Once we incubate the tissue with the radioactive ligand, it will start emitting radiations, in this case, beta particles. And once the beta particles are, or the radiation is getting emitted, we will expose a film or an emulsion. And what happens once the radiation falls on that emulsion or the film? It will now hit the silver grains in the emulsion and it will expose them. And we will see a particular kind of mark in those areas where the molecule has adhered or the radio labels have adhered. Okay, so this is how we find out our gene of interest, whether the transformants, genetic transformants, and so on and so forth. And the procedure is somewhat like this. First part is you need to label your cells with a radioactive compound. We discussed about the different types of radioactive compounds which we could use in biology. We could work with tritium, we could work with phosphorus 32, we could work with sulfur 35, or even iodine. So here in this case, the picture as is shown, you can see here they are using tritiated thymidine. We are using DNA. So thymidine is a base, and this thymidine is attached with tritium, which is a isotope of hydrogen. So once the cells are incubated in the radioactive compound, next is it is washed and then fixed. The reason for washing and fixing is unbound radioactive materials has to be removed. Fixing is to make sure that it enters into the cell and it stays there. Then comes embedding, dehydration and embedding of these treated cells onto a plastic mold or an emulsion slide or onto a slide. And once it is fixed, now you can immerse it in, dip it in to radiation sensitive emulsion inside a dark room. And then we can either store it in a dark box till further processing, or you can straight away go and process it. Uh, all these are done in the dark condition. And once it is processed, you can see silver grains over the cells or even the background. So we need background to be less and the cells to be more clear. So that should be our aim. So after the processing of the slide, we can see where exactly is our gene of interest or the DNA or our target sample is. So this is how we make use of autoradiography to detect the presence of radio labeled molecules. So this is again a diagrammatic representation showing how autoradiography works. So in the end, you can see that wherever there is this incorporated DNA, you can see a signal in the form of a mark or a band. Okay, an autoradiograph is a picture or on a film or a nuclear emulsion produced by a set of emissions of radioactive substances. So autoradiogram is a picture and autoradiography is a process. It is very widely used in detecting radioactively labeled DNA or RNA in a gel after separation by electrophoresis. And this DNA or RNA could also be transferred onto an inert membrane like nylon. That is by the process called blotting. You must have all read and learned about northern blotting, southern blotting, and western blotting. So, by transferring the DNA or RNA from the gel onto a blotting sheet helps us uh, in handling these materials during autoradiography. So mostly it is after hybridization, molecular hybridization, that we will be using autoradiography for detection. So in autoradiography, we can detect radioactive DNA bound to a filter paper during hybridization experiments. And whether the radioactive DNA is in a dried gel or on a membrane or on a piece of filter paper, nevertheless, whatever be the substratum the DNA is, it will emit beta radiation as long as there are radioactive isotopes embedded in it. The particles which turn regular photographic film black exactly the same way, when light falls on it, the photographic film gets black. So to see the location of the radioactively labeled DNA, a sheet of photographic film or an emulsion, as we saw earlier, will be laid on top of the gel. Here we are speaking about the gel after electrophoresis, or it could be on a filter after hybridization, and this could be left for several hours, or sometime it takes a few days for the radioactively labeled DNA to be completely visualized. And once the film darkens, we can see bands or spots in areas where our radio labeled DNA fragment was present. So, exposing the film must be carried out inside a dark room, especially to avoid visible light. And once it falls on our emulsion or the film, what happens is we will be getting high backgrounds, which we don't want. We want to see exactly where our radio labeled DNA molecule is present. So this is just to show how autoradiography works to detect labeled DNA or RNA after electrophoresis blotting. So he can see here after radiography, it will show you bands or patches where exactly your gene of interest is, because only to the gene of interest is the lags, the radio labeling is being done. So now you have your film showing the position of the bands. So this kind of experiments we could do when we are trying to isolate genes, we are trying to find out a particular gene, and by looking at this picture we can select the band which is carrying our gene of interest, and we could go for elution. So this is about autoradiography. Multiple applications in biology is attributed to autoradiography. As I said earlier, the technique can be used to determine the localization of the radioactive substance within a cell or a tissue. And this substance, once it is introduced into a metabolic pathway, could get coupled to a receptor or an enzyme or a nucleic acid. And the use of radioactive ligands makes it possible to determine whether the distribution of receptors in the tissue, both in vivo as well as the in vitro studies. For example, the ligands are generally labeled with tritium or iron 125, and the radioactive precursors of DNA or RNA, that is, we saw earlier, tritium. Thymidine, tritiated thymidine, that's what we call in the case of DNA, and tritiated uridine in the case of RNA. And these can be introduced into living cells and helps us determine the time of the different phases of cell division or cell cycle. So radioactive precursors of DNA or RNA could be made use of to identify or to see in which state of cell cycle is a particular cell at a time. Yet another application is it is possible to study hormonal production or metabolic pathways. And in order to carry out quantitative autoradiographic studies, it is also possible. And here we could use radioactive concentration standards, which is already available. And such standards are known as autoradiography standards. So we could plot a standard curve, and that can be used to estimate the radioactivity level of the sample. So autoradiography is a mechanism of detecting radio labeling. So a few advantages of autoradiography is minimal effect on radio labeling of biological activity, and direct comparison of biochemical and morphological data can be made use of with the help of radio labeled molecule. Then it prevents high molecular cross-linking induced by the gold conjugated probes. Probes are being used in hybridization processes where we could look for a particular kind of DNA or RNA. And sometimes this method of detection of radioactivity is found to be much more faster than the immunochemistry or the cryosections, which are being used widely to study various purposes like anatomy, the internal structures, and so on. So, or how enzymes react, and so on. So that is some of the advantages. So if we are listing out advantages, there could also be disadvantages or issues could creep up in autoradiography. Uh, First thing is that this is a long duration process which will consume a lot of time, and it also needs expertise. So, risk of diffusion of the product being localized into other parts or even people is one major constraint with autoradiography. Then there are something known as the disappearing grains. So, what happens is with time, the radioactivity could get decreased, and in that cases, it will be very difficult to detect. Okay, then of course, there is a risk of positive negative chemography, that is, contact between the sample and the emulsion might cause the direct reduction of silver grains and thereby preventing the process. So that is about when we are trying to make use of autoradiography for detection. Okay, then this is very highly sensitive because photons are radiations which generate heat and pressure, and this could probably result in particular potential backgrounds. So with electron microscopy also we can use autoradiography where high sampling and statistical analysis are needed though. So these are some of the issues, and overall and above all, we are dealing with molecules which emit radiation, and most of the times these radiations could be penetrating as well. And as far as we know, any external stimulus that is coming into the body will be considered by the body as an invader or a foreign particle. So nevertheless, the radioactivity or the radioisotopes, when they enter into the body, it has been sensed as a foreign particle, and definitely, and these penetrating molecules cause a lot of risk, lots of trouble. So let's check it out here. So we will be discussing more on radiation hazards. As I said now, radioactivity, radiation could penetrate into our bodies and could cause havoc. So before going there to check out the hazards and how we can, what are the safety measures that we need to follow, in the next session, we will be dealing about how exactly we can label biomolecules with radioisotopes. So we discussed about the detection and measurement of radioactive materials using Gigamuller counters, scintillation mechanisms, and then again, we discussed about autoradiography, a very powerful tool for detecting DNA molecules. With this, I stop here.